For almost every organism on Earth, biological death is an absolute certainty. Tissues degrade, metabolic processes slow down, and senescence eventually dismantles the body. There is exactly one notable exception to this rule: single-celled organisms. They manage to avoid a natural death by physically dividing their entire structure into two new, independent entities. Complex, multicellular life forms face a severe continuity problem. A mammal or a tree cannot simply split its body in half when its biological clock runs low. Because immortality is impossible at the organism level, life requires an active, overarching architecture to persist past a single generation. That architecture is reproduction. This scatterplot compares physical size against life expectancy. It's intuitive to assume larger organisms outlive smaller ones, but plotting a crow and a parrot breaks that assumption. Despite similar dimensions, they land on opposite ends of the vertical axis. The lifespan gap is massive. The crow lives roughly 15 years, while the similarly sized parrot can survive up to 140 years. This variance exists because lifespan is an evolutionary trait adapted to specific environmental pressures. It is not an automatic consequence of a body's physical dimensions or complexity. Ultimately, whether an organism survives for 15 years or 140 years is irrelevant to the long-term survival of the species. Reproduction acts as the universal equalizer, entirely bypassing the limitations of the individual clock. The most direct solution to the biological continuity problem is asexual reproduction. It requires only a single parent and produces exact morphological and genetic clones. For the simplest organisms, basic cell division is the entire reproductive sequence. Yeast generates clones through budding, while amoebas rely on binary fission. Higher plants also utilize asexual methods to bypass seed creation, a process termed vegetative propagation. This is achieved through specific structural units called vegetative propagules. These include runners, rhizomes, and offsets, like the structure seen here in the water hyacinth. The exponential speed of this cloning process is an incredibly potent ecological tool. The water hyacinth earned the title Terror of Bengal precisely because its rapid vegetative propagation allows it to quickly choke out standing water bodies, draining the oxygen and killing off the fish. While asexual cloning is an unrivaled method for rabbit colonization, it creates a fatal flaw. The complete lack of genetic variance leaves the entire population structurally vulnerable to a single new disease or environmental shift. To survive these shifts, many organisms that possess perfect cloning abilities will abruptly switch to sexual reproduction right before adverse environmental conditions arrive. This pivot is a massive energetic burden. The organism must abandon immediate, rapid expansion and dedicate its resources to building entirely new biological systems. The absolute prerequisite for this process is gametogenesis, which requires the parent organism to manufacture highly specialized reproductive cells. The process begins with a myocyte. This is a specialized diploid parent cell uniquely tasked with dividing to create independent, specialized haploid gametes. This table shows the precise mathematical reduction required during gametogenesis. The parent's chromosomal count is strictly halved from the 2N myocyte state down to the N gamete state, ensuring the eventual offspring isn't overloaded with double the genetic material. This precise cellular deconstruction is the required biological tax. Organisms pay this massive energy cost to generate the genetic variants necessary for long-term species survival. Creating genetically unique haploid gametes is only the first step. These cells are entirely useless unless they can physically locate and touch one another. The physical shape of these gametes varies wildly across taxa. Some algae produce identical cells called isogametes, whereas human reproduction relies on morphologically distinct male sperm and female ova. To deliver these cells, many species adopt a dioecious condition. This completely separates the male and female reproductive roles into distinct individual bodies. The physics of transferring these cells is remarkably consistent across most species. The male gamete is almost always motile, equipped to move, while the female gamete remains stationary and waits. These motile cells must cross hostile environments. They have to swim through turbulent aquatic mediums or survive being carried as pollen by unpredictable winds. The fail rate of this transit is immense. To offset catastrophic losses along the journey, males are forced to produce thousands or even millions of times more gametes than females. Gamete transfer is an extraordinarily inefficient biological gamble. It demands vast energy expenditure to facilitate just a single successful microscopic collision. The climax of this entire sequence is syngamy. This is the exact moment when two haploid gametes successfully fuse their structures, dissolving their shared wall to combine into one unified diploid center. There is a bizarre biological loophole called parthenogenesis. Organisms like rotifers and honeybees can occasionally skip syngamy altogether, developing into adults straight from unfertilized eggs. For most aquatic species, syngamy relies on external fertilization. This high-risk strategy involves synchronized mass spawning in open water, leaving the resulting offspring highly vulnerable to predators. Terrestrial organisms use an evolutionary counter-strategy, internal fertilization. The process of syngamy is moved deep inside the female body, protecting the vulnerable collision from the chaotic outside world. Whether it happens in a violent ocean current or safely inside a complex mammal, this cellular fusion creates the zygote. Syngamy acts as the universal reset switch across biological kingdoms. It restores the full genetic code and triggers the immediate construction of a new generation. The final phase transforms that single microscopic zygote into a fully functional organism. This is embryogenesis, a highly choreographed process requiring two simultaneous operational phases. The first phase is rapid cell division, or mitosis. This simply increases the sheer volume of biological building blocks. The second, far more complex phase is cell differentiation. Here, those identical building blocks begin organizing and differentiating into highly specialized tissues and organs. Embryogenesis is essentially the newly combined genetic blueprint automatically executing itself. It bootstraps a unique survival machine from a single dot of data. While this internal cellular logic is conserved across species, the final physical output mechanism varies depending on the organism's specific evolutionary path. Oviparous organisms export the embryo into the harsh environment, wrapped inside a calcareous shell. Viviparous organisms instead incubate their young internally, holding them until they are structurally sound to survive. There is a direct structural parallel in higher plants. The plant ovary develops into a thick pericarp wall to guard the embryo inside a seed. This cycle moves from the absolute certainty of senescence through the costly gamble of gametes and syngamy to the protective emergence of the final embryo. Returning to the lifespan data of the parrot and the crow, whether an organism is a simple cell or a complex vertebrate, its individual lifespan is nothing more than a temporary timer. Biological immortality at the individual level is a myth. But through the strict conserved architecture of reproduction, the genetic lineage successfully achieves practical immortality.